focus in on, the coded instructions. We know those coded instructions are now DNA. And how do these coded instructions, how do they specify certain traits? I'm just going to say traits. How do they specify traits? Okay. So we know these coded instructions are found in the nucleus, right? So I'm going to draw a cell here, and I got the nucleus here, and I've got these chromosomes kind of in the middle. Okay, and that's where the DNA is. Close that circle up. If you remember that it is the ribosomes, and that is way too big, but the ribosomes are actually outside of the nucleus. And so we have a little bit of a problem here. If I had only one set of notes and a student were absent, I wouldn't just give them my notes and here, take my notes and just have them, because I want to hold on to that, make sure that's preserved, that I don't have to do it again. But I might have them make a copy, maybe change it a little bit to fit their own wording, and then let them use those notes, the ones they copied, of course. So, the reality is, out here is a kind of a dangerous place for nucleic acids. We have these enzymes called that chop up DNA, and they get rid of this extra DNA. All nucleic acids in general, RNA included. Okay, so we don't want to expose them to that kind of environment and cause potential mutations. Okay, so that lies our problem. How do we get the instructions from the nucleus to the ribosome? Because the ribosome is where the action is happening. That's where protein synthesis takes place. So we're going to need an intermediate. We're going to need something to carry the message over. And so we're going to bring in a next player. And the next player is RNA. Okay, ribonucleic acid. So a couple things that are different between RNA and, let's say, DNA. Okay, let's just divide that in half here. RNA is made of ribose. That's the sugar. Or DNA is deoxyribose. It's actually missing one oxygen, deoxide. Okay. Another thing that we know is that this RNA is usually single-stranded. And you know already that this DNA is double-stranded. Okay, and last but not least, and certainly not least, it's the one you'll probably notice the most, is that there is a whole new nucleotide that washed up on the beach, remember? Washed up, okay. We certainly have A, we have C, we have G, just like DNA. Well, DNA we have a thymine, T. We have the new one, which is uracil. Okay, only found in RNA. A U. Okay, so it kind of replaces T, which means that A on DNA would match up with U on RNA. Okay, and I should also mention there are three types of these. Okay, there is what's called mRNA, which is messenger RNA, takes the message from the nucleus to the ribosome. We have tRNA, which stands for transfer RNA, and transfer RNA brings amino acids brings the amino acids that may be floating around in here and brings them over to the ribosome, okay, for assembling into a protein, right? Because amino acids are the building blocks of protein. And the third type we have is rRNA. Don't really have to know a lot about this other than that's what the ribosomes are made of. Okay. So I'm just going to give you a quick little rundown of what happens in here. Go with orange. And there are basically two steps. So protein synthesis, it has two steps. Step one is called transcription. Okay? And remember, we had to take the message from the nucleus to the ribosome without having the DNA leave the nucleus. So this is going to take and have DNA be copied into mRNA, messenger RNA. Okay? And I think of this like as if someone were a scribe for you. Let's say you broke your wrist, you couldn't write, you'd need a scribe. So what they would do is take what you say and put it down on paper. Okay? So this is transcription. Really, it's the same language. It's both nucleic acid. That's the same language, just a slightly different form. Okay, so we're taking this language. It's like in the example I gave, what you say is transcribed into written on paper, the same things. Okay. Step two is translation. Okay, and that's taking the mRNA and converting that into amino acid sequence, which will then end up folding up into a protein. And so here we are. I got to tuck down here. So here we are. We are at that point where we can talk about this amazing idea. It's like central. It's like a central idea, a main idea of genetics. They call it the central dogma. Dogma is idea. And that is this. We go from DNA to mRNA to a protein. Now we can also take DNA and make new DNA. So this process over here is that replication. This is transcription. And over here, this is translation. Okay? Main idea. This is the main idea that we're talking about. Okay. All right. Well, thanks. I hope this helped. And next we'll go into a little bit more of the details of transcription. Step number 1. Bye-bye.